I would like to say a few words about what is molecular diagnosis. Molecular diagnosis or molecular methods or tests used to identify a disease, predisposition of a disease, or monitoring a disease at molecular level. And this requires the analysis of either DNA or RNA of an organism at molecular level. Molecular diagnosis, in fact, combines the laboratory medicine with the knowledge of molecular genetics. And it has enormously revolutionized over the last decades, benefiting from the discoveries in the field of molecular biology. Actually, molecular diagnosis has transformed the medicine in many ways. Let us see how it has transformed the medicine. First of all, prenatal stage. We can know if the baby healthy, we can know disease predisposition. The patient is at this point. The patient is suffering from, then the translation, what should I prescribe, pharmacogenomics, what are the dose, and recurrence monitoring, how the disease has returned. So you can have answer of all these questions at molecular level by doing a molecular test, and you need a molecular test actually. And worldwide, if you can see that it is as of 2024, molecular diagnosis is more than $20 billion $20 billion industry worldwide, and increasing at the rate of more than 4% annually. So the transformation from traditional to molecular diagnosis is that the driving force behind the transformation is probably the rapid technological development in the field of molecular biology and our unprecedented ability to sequence the human genome. Though the transformation began in the virology world, it gradually expanded nearly in all medical specialties. That includes infectious disease, genetic disease, cardiovascular disease, cancer, and all these areas. Genetic mutations in ENT disease, that is what is the focus of this talk, of my talk today. So genetic mutation play a significant and increasingly recognized role in a wide range of ENT disease. These mutations can be inherited or can occur spontaneously. The key ENT areas that may affect are the nose and the sinus, the throat and the airways, and genetic susceptibility of ENT cancer. These mutations may be monogenic, that means single gene disorder, or polygenic. And new genes and mutations are published in scientific literature every week. So it is very difficult to say how many genetic variations are responsible for ENT disease. Actually, there is no exact number, but approximately I can say there are more than 2,000 mutations that is related to various types of ENT disease. First of all, I would like to say something about the P63 gene mutation. P63 gene
Suppressor.
actually it is a measure of it is a member of
P53.
P53 gene family and involved in the formation of epithelial tissues such as skin, teeth, bone, and the lining of the upper digestive tract. The location of the chromosome is chromosome 3, and the gene P63 gene is approximately 403 kilobase. That means this is a very big gene, 403 multiplied by 1000 base pairs. So, and it has 15 exons and undergoes alternative splicing from multiple to form multiple isoforms. You know, a gene is a segment of DNA, and not all genes are continuous. The big genes, they are called split genes, and their coding regions are separated by introns, and the coding regions is called exon. So this gene has, as you can see, it has 15 exons. So this is a very big gene. Most of the mutations found in P63 gene is a single nucleotide variation, that is SNP or SMP, we will say SNP, and very few are deletions and duplications. So far, more than 600 to 700 mutations, both as germline and somatic, have been identified, but not necessarily all mutations are identified. You know, mutation doesn't mean that it will create a disease. When a gene is transcribed or it produces from DNA to RNA to protein, so every amino acid is coded by 3 codons. So if the mutation doesn't change the codon, it doesn't have any effect on the protein. So it doesn't cause any harm. This is why not all the mutations are harmful. At least 40 mutations are P63 gene has been identified in people with EC syndrome. P63 can act as both oncogene and tumor suppressor gene depending on the splicing isoform involved. When genes are split genes, as you know that from DNA it is converted into mRNA, and in mRNA the non-coding regions are excised or removed, and they form mRNA, and this is called splicing. And when the fragments are joined, they can join in a very different way, that is called alternative splicing. So this alternative splicing can produce many different isoforms of the gene, and this P63 gene produces 2 main classes of isoforms. P63 acts as a tumor suppressor similar to P53, and delta N P63, the dominant isoform in basal epithelial cell. P63 can act both an oncogene and tumor suppressor gene depending on the specific isoform involved. In developmental syndrome, mutations are typically heterozygous, leading to a disruption of normal tissue formation. Well, as you know that we have in the human genome, we have 2 copies of every gene. So when there is a mutation in one gene, we call it heterozygous, and when we get the same mutation in both the chromosomes, we call it homozygous. There are some disease that affects the individual even if there is a mutation in one gene, then we call it autosomal dominant. And case, even if you have a mutation in a single gene, the individual is going to be affected by the problem. Developmental syndrome mutation, they are typically heterozygous behavior, disruption of normal tissue population. In cancer, it is not usually P63 gene that is mutated. Instead, the delta N P63 protein is overexpressed and blocks the tumor suppressor pathway. P63 mutation and ENT disease. Several rare genetic disorders are caused by mutation in P63 gene. Number one is ectrodactyl ectodermal the second one is AEC. I am not reading the whole name. And the third one is limb memory syndrome. So approximately 100 to 120 to 150 germline mutations of P63 gene are well documented to be associated with both EEC and AEC. Therefore, genetic testing of P63 gene is crucial for the diagnosis of EEC and AEC and related syndrome. This allows proactive multidisciplinary management of ENT, proper genetic counseling, and to prevent other manifestations from birth. Now, I just give you a comparison of P53 and P63. In fact, they belong to the same family, but its primary role of P53 is the tumor suppressor gene. Tumor suppressor gene means it prevents the formation of tumor, and P63 is the master regulator of the development. Common mutation type in P53 is loss of function, and in P63 there is a gain of function. And the main role of P53 is called the guardian of the genome and prevents cancer formation. And mutated, P53 is mutated in approximately 50 to 70% of the head and neck squamous cell carcinoma. And P63 promotes cancer cell survival proliferation. Association P53 sporadic cancer, Lee-Promeny syndrome, and P63 is poor prognosis in HPV negative aggressive HNSCC. The developmental role of P53 is not a major player in the development, but P63 is critical for limb, skin, and craniofacial development. Williams syndrome. Now I give you some idea of some disease which has a ENT relevance. Williams syndrome. Williams syndrome is a contagious gene deletion syndromes. That means it involves multiple genes caused by microdeletions of 1.5 to 1.8 megabase on the Q arm of chromosome 7. The region includes 26 to 28 genes, and the loss of a specific gene explains ENT-related manifestations. In the following table, you can see that there are several genes, elastin genes. Elastin is critical for the connective tissue and vascular elasticity. And next, there are three genes involved in craniofacial and neurodevelopmental phenotype. And LIMK1 or kinase implicated in neural development, and many may contribute to auditory dysfunction. STX1A involved in neurotransmission and may contribute to auditory processing issues. Niemann-Pick disease. Niemann-Pick disease is not a severe disorder but a group of inherited metabolic disorder. The gene involved are critical for lipid metabolism, specifically the transport and breakdown of cholesterol and other lipid inside the cell. There are three major subtypes, each of which distinct genetic causes. All forms of NPD is inherited as an autosomal recessive pattern. An individual must inherit 2 copies of the mutated gene from each of the parents. So, Niemann-Pick disease has 4 types, type A, B, C, D. Type A and B is the gene involved is SMPD1 and protein function it breaks down sphingomyelin in the lysosome. ENT relevance is sleep disordered breathing and snoring, sleep apnea, hypoventilation during sleep, and otitis media. And type C and C2, C1 and C2, the gene involved in NPC1 and NPC2 transports cholesterol out of the lysosome. Sensory neural hearing loss, dysphagia, and feeding difficulties in children. Tourette syndrome. Tourette syndrome is a rare disease caused by mutation
molecular diagnosis
important for primary cerebral
broad heading.
function. Over 40 genes so far have been identified. This mutation disrupts signaling pathway critical for brain, liver, kidney, eye, and ear development. So, you can see the genes and the ENT relevance is hearing impairment, abnormal breathing pattern, upper canal obstruction, speech and swallowing disorder, digital overlaps. Graves' disease. Graves' disease is an autoimmune disorder which the immune system produces thyroid stimulating immunoglobulins. These antibodies mimic TSH and bind to TSH receptor causing about production of T3 and T4. That means this is a form of, it causes a form of hyperthyroidism, and the gene involved is HLA, HLA-DR3, HLA-TP1, HLA-TP1, CTLA-4, and the ENT relevance is given here, and particularly in HLA-DR1 star 03 variant and HLA-TQA1 star 501 allele are strongly related to Graves' disease. These mutations predisposes autoimmune thyroiditis, which can enlarge thyroid gland, which is known as viter, causing airway compression, dysphagia, and neck swelling. Polymorphism in CTL4 polymorphism enhance the thyroid inflammation affecting the voice. PTPN22, the R6 PTPN22 CD40, it causes thyroid enlargement or orbitopathy ENT manifestation. FOXP3 leads to more severe thyroid autoimmunity contributing to tracheal obstruction. So, actually, there are hundreds of sorry, I have said thousands of genes that are related to the autoimmune disease. So it is beyond the scope of my discussion today to discuss every disease. So I have summarized the main key genes that is involved
Portal renal syndrome. Itu bronchial portal renal syndrome.
PCD, anesthesia resistance.
आगे select importance करते हैं primary ciliary distance cystic fibrosis paraganglioma
ENT related things. There is a chromosomal abnormality that also chromosomal aneuploidy that also affects the ENT disease. Chromosomal aneuploidy is very chromosomal aneuploidies are often associated with ENT manifestation either as a part of the syndrome, a syndromic feature, or due to direct effect on head-neck development. Chromosomal aneuploidy is very different from mutations or anything that happens in DNA. The chromosome is intact, but the problem is the numerical abnormality. And that means we have what we call 46 chromosome in our genome. In chromosomal aneuploidy, the number is changed. Either one chromosome is there is an extra chromosome, gain of an extra chromosome, or there is a missing chromosome, or may involve both autosome and sex chromosome. So autosomal aneuploidies include Down syndrome, which is most popularly known as trisomy 21, that is three chromosome 21. The incidence is 1 in 100. ENT relevance is recurrent otitis media, hearing loss, and obstructive sleep apnea. Here is the facial.
This is somatic
Edward syndrome, trisomy
Somatic.
18, that is
Down syndrome, adult syndrome.
The incidence is 1 in 3,000. Clearly, the annual obstruction, developmental delay, solving.
chromosomal abnormality.
Patau syndrome,
Klinefelter syndrome
is
আর হচ্ছে
trisomy 13.
ওই তিনটে
Incidence is
সেক্স
1 in
ক্রোমোজোম অ্যাবনর্মালিটি ক্লিনফেল্টার সিনড্রোম আর কি ডাউন সিনড্রোম ছিল না
clearly
One
facial abnormalities.
আরেকটা সিনড্রোম ছিল
Wrong.
compression.
47 ওইটা আর এটা হচ্ছে গাইড
And these are the external features. And chromosomal abnormality may also
Turner,
alter sex chromosome. Not
তিন ফ্রেন্ডের ছিল
chromosomes.
টার্নার ছিল
Sex chromosomal aneuploidy is
যেটা
mainly
জ্যাকবস
Turner syndrome. Chromosome that means that there is no
ডাউন
Y,
করতে হয়েছে অটোজোমাল
but they are
no,
actually
45 একটা ও 45
is
ছিল
1 in 2500.
45 ছিল না
ENT relevance is craniofacial anomalies, conductive hair loss,
টার্নার সিন্ড্রোমটা হচ্ছে যে হিজড়া হয় না হচ্ছে ফিমেল
obstructive
তিন ফ্রেন্ডের কাছে ওরা লম্বা হয় মানে মেল হিজড়া
Klinefelter syndrome, they have one
23
X chromosome.
ইয়ার্স
ডাউন তো 18
Incidence is 1 in
21 18
1200.
ডাউন সিন্ড্রোম
And
সিন্ড্রোম
the ENT relevance is high-pitched chorus
Equus.
in adolescence, reduced laryngeal prominence, androgen deficiency, and sensorineuronal hearing loss.
টিপুলেক্স
Then you have triple X syndrome. Triple X syndrome means they don't have any Y chromosome, they have 3 X chromosome. Incidence is 1 in
মূল হেডিং যেটা
1500, 1500.
স্যার
And
প্রশ্ন
the
করে
ENT relevance is occasional otitis media, speech and language
তো
delays,
দিবে না
craniofacial disproportion,
এতে
sleep disturbance, breathing problem.
মানে এটাই দিছে
And
ত্রিপুরা
Jacob
ইএনটিতে
syndrome, they have one
ফ্যামিলিয়ার ডিজিজ মানে
extra
এরকম
Y chromosome. They are also they are male.
Familial association.
100, 1000. They are basically very tall in stature, speech and language delays, otitis media, conductive hearing loss, and these are the phenotypes. And in Jacob syndrome,
এই এই উল্টা হয়ে গেছে না উল্টা হয়ে গেছে
problem
ফিলিং
is
সেটা ওইটা না
individuals
উল্টা হয়ে গেছে
হয়েছে
ছবি উল্টা হয়ে গেছে
the individual who has Jacob syndrome, it is found that they are sometimes engaged in social activities. Now, we have seen that there are many things employees that are responsible for the
স্মার্ট হবে
development of various different kinds of ENT problems. Now, what to start actually? Because how to, when you think that I would like to go for a genetic testing, what to start and what are the strategies of the genetic testing? That is the most important. The first practical challenge in implementing molecular diagnostics of healthcare practice is the issue when to genotype a patient. So there are two different approaches. Preemptive genotyping. Preemptive genotyping means genotyping or genetic testing of the patient for a specific gene or gene balance before any drug is prescribed. But probably you will not get my comment otherwise. Physicians are usually reluctant for preemptive genotyping because they think that, of course, when a patient goes to a doctor, the patient undergoes all kind of medical examination and other medical history. So the doctor may think that, okay, I know the drug, so I can prescribe the drug. But when the drug produces some kind of adverse side effect, then he changes the drug. So preemptive genotyping, in that sense, preemptive genotyping has many advantages. It can guide a clinician to select right medication, potentially improve therapeutic outcome from the outest, and cost saving by reducing influence and treatment associated healthcare expense, and improve patient safety, minimizing the drug reactions. This is one approach. The second approach is the reactive therapy. That means you already have prescribed a drug without genetic testing, and the patient has developed some kind of adverse reaction. Then the patient comes to you again, and you change the drug. So this is called reactive genotyping. Reactive genotyping is more like a and it comes of the suffering of the patient and delays underlying the timeline of the treatment. So this is the drawback of the reactive genotyping. Now, where to start? You either do preemptive genotyping or preemptive DNA testing or reactive DNA testing. But there are various steps you go step by step for the genetic testing. If you know the gene, this gene is related to the symptoms the patient has presented, you can go for a single gene testing. That means you can select the disease gene for the genetic testing. That is the first approach. But if you don't know the gene, and if you think that we have already seen that in ENT disease, in most of the diseases, multiple genes are involved. So, in that case, you have to order for a candidate versus panel genetic testing. That means you have to order for a gene candidate genetic testing. And next, if you do not get any answer from panel genotyping means you have selected few genes, 5, 10, 15 genes, and you do a test and you got nothing there. Then in that situation, what you can do, you can go for whole exome sequencing. And whole exome means in our genome, our genome is very big. It has approximately 3.2 billion nucleotides. Okay, and we have altogether approximately 25,000 genes. So exome sequencing means that you only sequence the coding regions of the genome, which accounts for only 1% of the genome. Okay? And from there, if you get any mutation or variation, then you are happy and you can prescribe a drug. Then if you still are in the dark, then you should go for whole genome sequencing. These are the 4 steps you go one by one for the diagnosis of the disease. So candidate drug versus panel genotyping.
কিছু হচ্ছে
Gene panels are crucial tool in molecular diagnosis, allowing testing multiple genes. I've already discussed, and when we do panel genotyping, a disease can be related to multiple genes. Conversely, a particular gene may be associated with multiple disease. Selection of gene panel is based on extensive review of medical literature. But there are a group of people who are actually ready to take this. So they have actually designed various gene panels for various diseases. So you can choose a panel from those design panel for genetic testing, or you can choose and pick and design your own panel or customize your own panel and go for the genetic testing. That is one approach. Whole exome sequencing is a technique that sequence all the protein coding genes in the genome. In recent years, it has gained attention as a promising tool in diagnosing genetic disease, which provides a broader view. It gives you a big picture of the genetic situation. So the exome of human consists about roughly 180,000 exomes. Okay? It contains about 1 to 2% of the total genome. Whole exome sequencing targets only clinically relevant regions, thereby avoiding vast non-coding data in the genome. But in our genome, in non-coding, approximately 98% of the genome is non-coding. That means they do not code for any functional coding. Then why nature has put so much of DNA in our genome? That is a very big question. But in these non-coding regions, we have many different kinds of elements like regulated sequence, repeated sequence, pseudogenes, and all these things.
দিয়ে যে ভিডিও করলাম
Sequence or repeated sequence. So regulatory sequence, they actually regulate the expression of a gene. That is why introns sometimes also play an important role in the expression of the gene. And why would you go for exome sequencing? If a patient shows unusual or unexplained symptom, sensitivity that cannot be explained by genetic tests. Whole exome sequencing can help identify rare or novel variants in related genes. It can also help find new candidate genes or variants in population not well represented in many existing panels. Actually, there are a lot of databases where you can find which gene is related to which disease. I'm sorry, I forgot to put this slide in here. You can go to ClinVar, FarmVar, ClinPGX. These are the web resources. You can visit these web resources when you have free time, and you will get a very good picture which and there is a search engine here. You can either put the name of the disease or you can put the name of the gene. and then it will tell you this gene is related to this disease or this gene has a this variant and for this disease you really need to go to for genetic testing. There are 4 different kinds of plans. Genetic testing required, genetic testing recommended, those related, and another one is there is no significance. Whole genome sequencing is the most comprehensive approach of molecular diagnosis, but it is not the first line of choice for most patients. It is recommended when there is an unexplained symptom presentation in patient under investigation. But you actually in some kind of time what kind of disease the patient is suffering. I really don't know. Captures non-coordinary sexual whole exome sequence will miss this kind of variations, non-coding variants, structural variants, and cognitive variations. Though it is often it offers several advantages and bridging gap in information often from panel-based genotyping or whole exome sequencing, there remains many practical challenges from its routine use in clinical practice. What are the challenges? Whole genome sequencing generates massive amount of data. So you need the help of another group of people who knows computer and also a little bit biology. For these people, they are now called bioinformatician. So you need a bioinformatic analysis of the whole genome data to find out what is the message conveyed from the whole genome sequencing. And whole genome sequencing are classified as pathogenic, lightly pathogenic, variant of unknown significance, and there are 4 types. Finally, in the report, they say this variant, we found this so variant, this variant is not pathogenic, this variant is pathogenic, this variant is not pathogenic, this variant is lightly pathogenic, and this variant is variant of unknown significance, and this variant. So, whole exome sequencing remains more expensive than I think nowadays it will be at least approximately 1 so this is very expensive. So molecular diagnostic testing methods. I'm not going to actually, I have just put the names. These are all standard methods. These are actually related to the results, samples, sample percentage. So you can use PCR and there are a lot of variants of PCR. PCR are related to ASPCR, PCR, So, the technique testing, if you want to go out to the technical testing, the laboratory decides which method is going to and for
শব্দ কিসের
guidance and opioids, there are several methods. Number one, this is the conventional daguerreotyping, but this method has some limitations. But for instance,
জিন এক্সট্রা
this method has a longer analysis time, maybe several weeks. Liver intensive and highly dependent upon the experience and the skill of the analyst, and this method cannot be automated. Then fish. Fish is the the result really depends on what kind of probe the lab has used. And fish also requires to disposable evaluations and transformations.
It is not possible to hear anything.
So this
Please tell loudly, sir.
सबसे सामने
We can't hear you.
ये तो सबसे सामने मसले चल रहे सबसे
We can't hear you, sir. Please deliver this loudly.
Also helps study how patients functionalize therapy, predicts disease progression and thanks for your attention.
Thank you, sir, for your nice and elaborate presentation. It's a rainy evening, and topics was quite difficult. So it's a sleepy environment is going on here. So now I would like to request our Department Assistant Professor, Dr. Anisur sir, to give a short speech. Anisur sir.